Optimal health for high performers. This is the Health Upgrade Podcast with Dr. Nawaz Habib.
Hello, everybody. Welcome back to the Health Upgrade Podcast. This is Dr. Habib. I am here with JP. How are you, sir?
Doing very well, thank you.
And we are honored to have our guest today, Dr. Sabine Hazan. How are you, Sabine?
I'm doing good. Thanks.
We are excited to chat gut health with you today. You are, for lack of a better term, the queen of the BIF. And we'll get into what that means for most people. They don't truly yet know what the BIF is or Bifidobacteria overall. But what's really unique is your focus is very deeply on optimizing the gut microbiota and understanding gut testing, understanding what needs to occur to optimize that gut bacterial population, and ultimately saving Bifidobacteria because of its important role in so many different parts of our overall health. I want to first get a quick sense of how you got into this particularly. I know gastroenterology is something that you obviously leaned heavily into through med school and post, but I'd love to get a sense of how you became so enthralled with the microbiome in itself.
I think it starts, I'm a woman born in Morocco, so always had a chip on my shoulder, not being heard or challenged, right? Like you can't be a GI, you can't look at the microbiome, you can't be a doctor. So all these can't over a lifetime have kind of like developed this warrior in me that my two favorite words are watch me. So it was basically that chip on my shoulder that really got me moving into this direction because, I started off, like you said, in GI. And GI, I went into GI because some guy said, we don't take women in GI. And I said, well, I'm going to be the first one. And I was the first one at University of Florida. And then, from there, went into the clinical trial route, really to always see something that doctors weren't seeing. Like, what's next? What's coming? Like, okay, we know we're treating Crohn's with this drug, but what's next? What's better for my patients than this drug, right? So always being ahead of everybody in the clinical trial world. When the clinical trial world became fecal material in a capsule, and I had all these questions, one could say I went rogue. And I think that's my inquisitive mind, my, not accepting, I think, I was born a scientist because a scientist is always challenging the dogma, always questioning why, and not accepting that there's a foundation and accepting also sometimes that the foundation that we were taught may be wrong, right? So I went into the microbiome space because I was seeing something when I was doing fecal transplant, the process of taking stools from a healthy donor to an unhealthy, but I wasn't reproducible, right? So it was like, well, this donor helped this patient with Alzheimer's, but why am I doing this on this other patient? It's not working, right? So it was really trying to see, why isn't it reproducing? Colleen Kelly did a beautiful study where she showed 2 patients with alopecia areata that grew hair after fecal transplant. And I was more interested in what are the microbes that did that than can we bring a fecal material product to the market? Because I tend to think we push material products here and there, but we don't sit down. And believe me, I've done 3 decades of clinical trials, so I see what's going on in the clinical trial world with pharma. We push these products that give like 20, 40% success. But then what happens to the others?
Relative risks, not even absolute risks.
Correct. And then but what happens to the rest of the patient? Are we supposed to just, let's give everybody this pill? So that's why I think, the microbiome to me was really, how do we analyze? And then seeing the corruption in the field of people pushing probiotics, people, and any probiotics, it didn't even have, Bifidobacteria in the probiotic and they're selling it, or people pushing these tests that mean nothing to the general public and worse, means nothing to the doctors, right? I remember getting a test from a company that was, that's bankrupt now and was essentially indicted by the feds, Ubiome. And I called my buddy, Neil Stolman in San Francisco, and I'm like, what is this test? What's Bacteroides vulgaris? He goes, heck if I know. I'm like, well, if you don't know and I don't know, we have a problem, right? How is the patient supposed to know? And what are they supposed to be doing with that data, right? And so the first thing that I did was look at all these tests, send my stools, realizing, these tests are just not valid. And in fact, Scott Jackson at the National Institute of Standard did a beautiful study where he actually looked at 9 different labs and showed that none of those labs are reproducing the data. Like, he created a microbiome consortium and he gave it to different labs and he got different results. And he got different results at the genus, but imagine at the species level. So you're a doctor and you're basically getting a patient, and your patient has E. coli based on the test. Are you going to treat that? Are you not going to treat that? And that's the problem with all these tests, is they're not valid, verified, and reproducible. So if it tells you that it has H. pylori, well, that's a PCR. Does that mean the patient was releasing a portion of the H. pylori and therefore never really had H. pylori because he's cleaning it out? Or does it mean the patient have H. pylori and is at risk of stomach cancer? So, and then what do you do with that information when you see a fragment of H. pylori and you believe that you should treat the patient, and now you just demolish the microbiome that he was trying to hold on to because you were going after a piece of a microbe that's really not important, right? So I think that all that I saw the corruption and I just had to do something about it. And then, like I said, I spoke at Congress last week and I basically said that, it was basically, I felt like I was in a unique position. I'm treating Malibu patients, some of them are celebrities, some of them are billionaires. And I own a genetic sequencing lab that looks at the microbiome. And for the most part, I feel like we have a validated assay. To me, validation is like seeing your microbiome, being able to figure out this is you out of a stool sample. If somebody blocks the toilet in my house, I know who it is. So, that was basically, I was in this unique position of, I have a genetic sequencing lab. I have a portal with the FDA that does clinical trials for pharma, and I'm a Malibu physician. So, I started collaborating with a lot of doctors. You know me, I'm a team player. I call Dr. Brody. I call, all these doctors, Peter McCullough at the beginning, Pierre Corey. And, it's important to come together on this. So I think at the end of the day, what am I? I think I'm just a rebel. And I think I'm a girl that doesn't like to accept the dogma. And if I see something, I speak because we have to.
We need rebels in the field. So I'm glad that you're here.
One of my friends is head of academia in a very prominent institution. And I said to him, in 2026, it takes bravery to be a doctor. And he goes, it does.
Yeah, no doubt. I want to ask, how do clinicians and patients that are advocating for themselves, for their own patients, how do they figure out which tests to run? Because microbiome tests are at this point what seems like a dime a dozen, right? And there's PCR tests, there's shotgun tests, there's so many different types of analyses. How can we become discerning practitioners to determine which is the best option for us?
Well, I think the key is basically to have validation, to have clinical data, to have reproducibility, and to be righteous to say maybe, maybe not. At best, the microbiome is just a research tool. This is why we enroll patients in our clinical trials and we tell them this is a research tool. We may be on the right. Listen, I've been pushing the Bifidobacteria and save the BIF. I could be wrong, right? I mean, we're in trillions of microbes. We have no idea. There could be another. I was just, reviewing something today about animals in the ocean. And there's this particular animal that, is called an immortal animal, right? What's in that animal, right? So these are the questions we have to understand. And I think if you start with the foundation that we don't know the microbiome, we don't know sh.d, I think it's easier to see it. And if you start with the foundation that we don't know anything, I think it's easier. So patients, unfortunately, it's too early to do anything because I have people calling me and saying, oh, can you fix my Parkinson's with fecal material? Can you fix my Alzheimer's with fecal material? At best, we have a signal. So Progenibiome, which is the lab that I created, which is a research lab, we're seeing some signals, right? Signal of what Parkinson's could look like, signal of what Alzheimer's could look like. Now it's important to work with other doctors that are practicing the art of medicine, that are doing something else to see, am I right? Did the signal change, right? I have a signal of what I believe is a healthy microbiome, right? And I have a signal of what I believe Parkinson's is. Did that signal change when I gave, vagal nerve stimulation? Did that signal change when I gave this protocol? Is that how we're improving? How am I doing it with autism, right? So autism is kind of like, I took that disease, first of all, because it was kind of serendipitous, it was divine intervention. This whole field for me is divine intervention. Really, God has guided me. God has trained me as a warrior through my life. All these roadblocks that I faced as a doctor, as a practicing physician, as a medical student, were all the training from God to be allowed to speak right now, right? And so to me, this whole path was divine intervention. When I improved a case of Alzheimer's by fecal transplant, one patient, N of one, it took me a long time to publish the data. But I went to Dr. Feingold, Sidney Feingold, who's the top guy, was the top guy, infectious disease, worked at the VA, wrote the book on anaerobic infections. And I went to him and I said, what am I seeing when I'm improving Alzheimer's? And he gave me a paper and he said, you're seeing these microbes. When you get your device to analyze the microbiome, you're going to prove me that I'm right because you're going to find these microbes, okay? And he tried to push that finding of that microbe with all sorts of pharmaceutical companies. Nobody was interested in that. I took that paper, put it in a safe, and I said, okay, well, I guess I'm going into the microbiome business, right? So I bought the machine. It was kind of a turn of event. The first patient, when the machine was installed, first of all, I got this scientist, Dr. Papuzzi, who basically was behind the BRCA gene, brilliant scientist. And I said, I need an assay that's valid, verified, and reproducible, okay? And I said, I want to look at the microbiome before and after fecal transplant, right? So that I can kind of understand. And then he said to me, okay, let's embark on that. And at the same time, the same week that the machine came and he came, I had a 17-year-old with autism in my office, banging his head on the wall. And autism was the patent of Dr. Feingold and Dr. Barodi to use. Dr. Feingold came up with vancomycin and Dr. Barodi came up with microbes, right? And so fecal transplant. And so the two of them, it was, I was working, on both levels. When Dr. Feingold passed away, Dr. Barodi entered my life and we were talking every night, trying to figure out, wrap our heads around the microbiome. And he would say, to look at the microbiome is to see the power of God, because all these trillions of microbes that are just connecting together and how we're all different and yet we survive and have different traits thanks to our microbiome. And these traits are important to develop a certain, mission in the world, right? So when you look at different cultures and how together that diversity represents health of the planet, health of humanity, health of the microbiome, how diversity in the gut is health and how loss of diversity is disease. So we basically started talking all that. And so with that kid with autism was the beginning, right? And what I did with autism was really try to figure it out. And how did I figure it out? Well, I stepped into COVID. I understood the microbiome of newborns was high bifidobacteria. I saw the microbiome of autistic kids, low bifidobacteria compared to their siblings, compared to their, to the rest of the healthy population. And then how, when you restore the bifidobacteria by suppressing the toxic microbes and allowing the field to grow good microbes, these kids resume speech, right? So that's the same thing that I want to bring to other diseases, right? It's not, but here's the interesting thing. Autism is loss of bifidobacteria at first because it's simple in a way, because we're comparing these kids to newborns that have 90% bifidobacteria, right? So what about other diseases? The microbiome changes with aging, right? There's a different groups of microbes that start increasing that give you that resilience, right? Are you missing those microbes that gave you that resilience, right? Think about bifidobacteria. Bifidobacteria starts as an infantis, right? The newborn bifidobacteria, and then develops into the adolescent, into the longum, right? So all that, even that transformation, that loss of bifidobacteria allows for resilience by growing other microbes. So what happens when you take an antibiotic and you kill off those microbes that you built, right? So think about it. I'm the girl that was in Morocco. I built up a resilience to be allowed to be in America, right? To be exposed to this and then I've killed a lot of microbes that I had from my environment in Morocco, right? I've lost a bit of that resilience, right? So how do I build up that resilience, right?
So there's some recent research, and it's not at my fingertips as to who published it, but seeming to show that the microbiome is very much shaped and controlled by what your genetic makeup is. That there's a very strong control of microbiome by genetic influences, as opposed to what I think people generally consider to be more of an environmental thing. What are you eating? What environment are you living in? What have you done? And I'm not exactly sure how they did that research to show that, because the microbiome is trillions of different microbes. With human beings have 20 some odd thousand genes. There's literally millions of genes in your microbiome. So I'm not exactly sure how they did the research, but how does that play into things like taking supplements for your microbiome or doing a fecal transplant? How does that play in? Because you would think that the genetic code's not changing. The epigenetics are, but the genetic code isn't changing. So how long do those changes last before the body just returns to where it was before?
I think that's a great question. And I think, we just don't know. And I think, when people do research, it's important to reproduce the research. But here's the problem. In the microbiome space, it's very difficult to do very good research because there's so many factors, right? So when I'm comparing a population of Parkinson's patients, for example, am I comparing a population that's uniform, that's in the same family, that have the same genetics? Am I comparing a population, from Africa versus a person from Africa that lived in America? Am I comparing to an American from Nebraska? Am I comparing a vegan to a vegetarian to a carnivore? Am I comparing someone that takes lovastatin? So it's very, very complex to do that kind of research. And let me just say, that is the biggest challenge we have. And it is, and people have said to me, well, you could just put it all through AI and it'll figure it out for you. No, it will not figure it out for you, because unfortunately, there are things from a questionnaire that people do not give you. So for example, I'm analyzing two people, right? And I see one guy is off. I'll go back to that patient and say, do you drink alcohol by any chance? Because it seems like there's a destroyed microbiome that I see with alcohol, for example, right? It's going to take me a while to get that history of alcohol, right? Because most people are like, healthy. Do you know how many people put on their history and questionnaire they're healthy and they have like MS, Parkinson's, autoimmune process? I'm like, that's not healthy. You have a past medical history. Or even, I'm healthy, but yet I'm having digestive problem. You're not healthy. I'm healthy, but I'm having skin problem. You're not healthy. So I'm healthy, but I'm losing my hair, or my nails are brittle. You're not healthy, right? So I think it's that art of medicine to capture the history that AI will never be able to capture, in my opinion. Because in order for humans to open up about their trauma, the fact that they're using alcohol because they've been raped or been, suffered some losses in the family, right? It takes the history, it takes the trust, it takes that patient-doctor relationship where you gather the history. So any research that's done on the microbiome right now is most likely flawed. So I would take it with a grain of salt, because I always look at this data. There's people coming out, scientists that work in the lab on mice and rats, and they come out with, we've discovered the microbiome of MS, right? And I look at that and I read it and it doesn't match what I'm seeing. So I always tweet back and say, go back to the lab, guys. I'm not seeing this, right? There was and I'll give you an example. There was a study once that showed a certain microbe for Crohn's disease, okay? And doctors went wild. Oh my God, we found the microbe for Crohn's disease. It turned out it was a microbe that caused dandruff, okay? So was there contamination of the samples? What happened there, right? So you have to be in a world of the microbiome. You really have to be righteous to say, I don't know. And then you have to say, I'm willing to look at it, but then I'm not sure about what I'm looking at. But let me try to have the courage to give this product, right? And let me see if it works, right? And then when you achieve, I always say, to understand the microbiome, you have to achieve a cure. To achieve a cure, you have to practice the art of medicine, and you have to be brave. Very difficult to do in 2026 when you've got the media suffocating you and censoring the research, and when politicians are giving advice and celebrities and influencers are giving advice to patients on what to do. It's very difficult to go against that current, against that narrative, and to kind of stand there and say, okay, you're doing this for COVID, but I just don't see it the right way. I think we should go this way. It's very hard to convince people when everybody's going one way to tell them to come this way, which is the uncertain route, right? So, genetics, which came first? The microbiome, the microbes, or the genes, the DNA? Which came first, the chicken or the egg, right? Which comes first, the peptide, the protein, or the bacteria, right? So, we're just all made up of protein, right? Our DNA is a bunch of proteins. So what came first, right? What's changed, right? So yes, genetics. I mean, I personally think that it's the two of them go together. That's why I was so not opposed, but I was questioning the whole fecal in a capsule, because we don't give blood transfusions in a capsule. We don't give blood in a capsule. We don't give hemoglobin in a capsule. We match donors, right? We look at the type of blood and we match donors. And then we look at the blood and we say, there's no hepatitis C, there's no HIV, there's no Hep B, that blood is clean, to the cleanest that we can find. Now, I will challenge to say that there are more microbes in the blood that we haven't figured out that when we give a transfusion probably start a disease process. But, this is like where the research needs to be. So, but it cannot be if we are still, saying, okay, well, this is genetics, and therefore let's just put all our eggs in one basket and it's all genetics, or this is all microbiome. I'm trying to get us to both.
Or that the microbiome, because we're spending a lot of time talking about the microbiome of the gut, but there's a microbiome of the airways.
And they're all interconnected.
Square meters of airway. You've got the skin microbiome. So there's
and I propose to say they're all interconnected.
Oh, 100%.
Yeah,
You take a nose sample and you will notice the microbes. Like, let's say you grow E. coli in abundance or Staph aureus, you will see it in the gut. So anything you put on your skin, in your nose, in your mouth, you'll see it in the gut.
most hospital-borne infections after surgery are actually caused by the patient's own nose.
Yes.
I mean, it's more than 50%. It's like 60 some odd percent of these infections are caused by the patient's own nose. Not that they're like touching their nose and touching their wound, but it's just part of the air system.
Well, because the nose has blood vessels and lymphatics and nerves, and those circulate, the blood vessels circulate. So if you have a bacteria in the nose, I propose to say it's because we over-sterilize. We give so many antibiotics when we're to patients in the hospital that we're sterilizing the gut, so the gut doesn't even recognize a simple microbe that's in the nose. So, and that's the direction that humanity is going, and that's exactly what I was going to say. Bring it back to, Pasteur and Antoine Béchamp. Is it pasteurization? Is it is sterility, or is it the terrain, right? I propose that it's both, it's that balance, right?
Yes.
But we don't understand as doctors balance. We don't understand that if you're killing the tumor, you need to optimize the gut to bring back the balance. We don't understand that if you're giving an antibiotic for urinary tract infection, you got to optimize the gut so that it doesn't destroy, so.
You've mentioned alcohol, antibiotics as two of the bigger culprits in knocking down the gut microbiome. And you've also talked a little bit about how when people are born, it's the Bifidobacter infantis that is, in fact, the largest component of the microbiome. And then more research is showing that the population, as dynamic as it is, shifts into different populations at different ages and different stages of life. Not a number age, but rather life stage, playing huge roles in neurodevelopment when we're younger, in so many other important factors of being healthy, fertility, et cetera, as we get older, and then basic changes in menopause and whatnot for females and, cardiovascular disease, huge changes. The big finding that you had, I think, early on was that Bifidobacter was as important as it was on the development side. Are there any newer findings in terms of that bacterial population as we go through those stages that are showing up as being as important?
So it's interesting because Bifidobacteria is not as important, for example, in Parkinson's, right? But it is important. We're seeing it, loss of Bifidobacteria in Alzheimer's, in autism, in Crohn's disease, in Lyme patients, in
Schizophrenia?
long COVID, vaccine injured, mental health, anxiety patients. Think about COVID. Patients were super anxious. Their Bifidobacteria was 0. Their Bacteroides was super high. It's also decreased in bipolar disorder, right? So now there's other microbes of bipolar disorders that come into play, lost microbes. I think, we tend as society to just look at the negative, like, oh my God, E. coli is up. But we forget the positive, what is lost, right? Part of the reason that I focus on Bifidobacteria is because to me, that's really the microbe that's disappearing. If I were to say, what happens with disease? You've killed off your Bifidobacteria first, and then disease process occurs, right? It's kind of like, and I bring it back always to blood transfusion, right? Your hemoglobin is 6, you're at risk of a heart attack. The heart attack wouldn't have happened if your hemoglobin was normal, right? So you always want to kind of keep up on top of the hemoglobin to make sure your patient is not bleeding because the consequence is the heart attack, right? So it's the same thing with the microbiome. I think, in my opinion, and that's just my humble opinion, but I think it's been reproduced by a lot of doctors and scientists out there, and I think a lot of people could see it. You kill an important microbe that helps break down the food to take the sugar and put it into the cell, you have a problem. Your cells are not going to metabolize. Your immunity is going to go down. You're not going to have the energy to get out of bed. So it's a very important microbe. Now, I'm not saying there's no other microbes that are doing that. Obviously, there are other microbes because they all come as a gang and it's like, oh, Bifido is down. Let's bring on this microbe and this microbe to do the same thing. But I think to me, I focused on the Bifidobacteria because that was kind of the beginning and that was the most obvious one, mainly because it does appear in the children at a higher level. So
It makes sense.
to me, that was the beginning. Is Bifidobacteria a microbe of Americans because we're used to drinking milk and cow milk and human breast milk? Or is that the microbe for the Chinese, for example, or the Chinese population that has a completely different diet, right? Is it a microbiome of, people in Dubai, right? Well, Dubai is actually a very melting pot, right? It's like, to me, it's like, so many cultures, so many, they have such a small area with such dense population that it's all mixing. So it's interesting because even the food is kind of like, not, is over processed, right? So Dubai is on its own. But if you go to like Zimbabwe, for example, and you go to, the mountains of Afghanistan, for example, or you go to Morocco to the little Bedouin villages, it's a different microbiome there. And I don't think Bifidobacteria is necessarily what we will see or find there.
Which is fair enough. And I think the microbiome signature for me, that's interesting, and particularly in North America, because this is where you've been testing and you're aware of this. But it almost feels like based on that signature, we're able to determine resilience, right? And resilience for me is kind of this ultimate goal of how much can your body handle from an allostatic load standpoint, and how capable is it on bouncing back? Can we bounce back through autonomic regulation, through autonomic flexibility, back to a homeostatic baseline? And the biochemical stress and whatnot of the microbiome is going to play a huge role here. And the pattern that you've identified is that generally when we're younger, the bifidobacter level is higher, which is linked to, or in a healthy population, a higher bifidobacter, a higher rebound ability, or a higher ability to rebound from stressors. Versus when the bifidobacter level is lower, we have, and it shows not just in autism, but so many conditions, as you mentioned, that we're not able to bounce back, and these conditions tend to occur. And so would you say there's kind of that style of correlation is what we're looking for when we're identifying the changing population?
Yeah, I think, if anything, the pandemic has kind of, shown in a way, and at least it showed me a pattern of resilience, right? And that's resilience on both sides, right? Resilience on the person who took a vaccine and resilience on a person who chose not to take a vaccine, right? Because, some people got affected from the vaccine, some people got affected by COVID, so it's resilience on both ends. I think on the whole, the population has the ability to be resilient. I mean, think about it. If you're eating those nuts in New York, that are out there exposed to the environment, you're pretty resilient. If you can step into a McDonald's and eat the French fries that have sat on the counter and the same machines that are being used, that's a demonstration of resilience, right? I think that's what we should aim to have. I think we tend to be over sterile. And that's, again, where I'm bringing back the idea of it's not about Pasteur or Béchamp. It's in between. Yes, you don't want to be going from a sterile environment to a mud bath where, pigs have done their thing, but because you're going to get sick or go into a pond that is infected. But at the same time, over sterilizing stops you from even being exposed to a pepper. Most people don't realize a tomato and a pepper have viruses, and, if you're overly sterile, you're not going to be able to tolerate that pepper. And is that why people are allergic to certain foods? Maybe.
Well, there's a lot of evidence that childhood allergies that we're seeing are a function of the over sterility that we're exposing our kids to or not exposing our kids to, and that some of the old advice that doctors used to give of not exposing kids to peanuts until they're 5 or 6 years old, or not to eggs or other things, actually leads to higher levels of these allergies, and that you actually have to, in populations where kids are literally weaned off peanut containing products, have very low risks of having those diseases. Or those
absolutely. I think we overdo it. I think we overkill from, we take these beautiful babies with 95% bifidobacteria, and yes, they're going to survive getting 1, 2, 3, 4, 5 vaccines, but then we give them 72 vaccines, and then we give them, and then they get an ear infection because now you've just killed off their microbiome during that time. Now we're giving them antibiotics. So they're trying to climb up and they're trying to survive, but we're suppressing them again. So I think we need to, think about the babies that are born in a sterile environment. And I have those 2 kids, by the way. I have that kid that I, raised sterile to the max, cleaned up all the bottles. And then I have that kid that I raised just eat from the floor, right?
Second child.
He's a second one.
Let me tell you, the second one, I overdid it with my first one.
Yep.
But we as parents tend to be so over the top protecting these kids. We panic and we overdo. We forget that they have their own little immunity. They have their own little resilience. We need to let them fall for them to pick up, right?
We forget that 50 years ago or 100 years ago, they were crawling around on dirt floors. And
yes.
They were, and 1,000 years ago, they were, God knows what they were doing. And 10,000 years ago, they were in a cave, in a puddle. So,
absolutely. And you can see that, by the way, when you go to the areas of Africa where, they're still hunting for the zebra, and they're eating the zebra, raw, and they're full of blood, and then they're touching their kids, and they're touching the dirt, and then they're eating the honey from the beehive directly, so you could see that, all those microbes intermingling, right? You could see that in the Amazon
And
jungle.
they're the healthiest.
Women.
And they're healthy.
Yes. I mean, look at the
Absolute health.
Amazon jungle. We don't have heart disease in the Amazon unless they intercepted the chips and the Coke from America and the products of America. Then yes. And by the way, now the big disaster in the Amazon jungle is they got Wi-Fi. So all these kids are brainwashed to be like American kids. We're the worst as far as education.
We are exporting bad stuff.
There's my daughter did a study with a professor at USC on the island of Kiribati. And in the island of Kiribati, what was happening is if you look at the island, they have an increased rate of obesity and early mortality and diabetes. So obesity, diabetes, increased rate of mortality. And if you look at what happened in that island, it's really because we changed the diet and we gave them chips and Coke. They would drink a gallon of Coke and chips all day. So of course they're increasing. They're not hunting. They're not farming. They're not eating the fresh fruits and vegetables from the environment, from the island. We're poisoning them. So, and then we say, oh, you know what? You're having diabetes. Let's give you these meds. And you're getting infections because your microbiome is out of whack because you're overweight. And let's give you more meds. So we, pretty soon, it's going to be a disappearance. So my fear is if we don't stop, we're killing humanity. I mean, I can already tell you. So my big passion project, and I'm hoping I'm going to have somebody's going to want to fund it. But my big passion project would be to map the microbiome of the world to basically preserve the microbiome, to see what does the Amazon. I have a few samples here and there, but it would be great to have a bigger, sample size to kind of say, this is what Amazon looks like. Oh my God, they've lost microbes in the last 10 years. How do we restore those microbes, And kind of map the population, because I fear, my biggest fear as we are seeing the disappearance of Bifidobacteria is we're going to lose. Look, lactobacillus, it's very difficult to find it in humans. I mean, I have 4,000 samples. I think one patient has it, So is that why the majority of people cannot tolerate dairy? Because they've lost the ability to break down the dairy to get the calcium in the cell?
I
don't know.
The gene's only been around for 8,000 years, so past childhood. So it's still.
Yes. So I don't know. I mean, it's interesting too, because I was telling to someone's like, well, we're living longer with all these sterility. And, if you do believe in the Bible, right, and you do believe the stories of the Bible, you'll understand that Noah lived to 950 years, supposedly. Did he live to 950 years because he had animals 2 by 2 in his ark and therefore was exposed to animals all around him? I don't know. We'll see.
There are historians who think that there was a translational error between months and years. So that 950 may have just been months, which is still a decent life. I know it's in his 80s.
In those days, with all these microbes. But somehow they became resilient to, and again, what happens to a man that's living with a bunch of animals, right? When you look at the farmers, other than the farmers that are destroying their guts with alcohol and drugs and all that, but when you look at good farmers that practice regenerative health and regenerative medicine, they're usually, I know a lot of farmers who have regenerative farms who never got COVID and never got vaccinated. And I tend to think it's probably being exposed to all that. And, it's so funny because my argument on the whole sterility versus microbiome, Dr. Feingold lived to 97 years old. He was collecting stools of patients with Alzheimer's and autism and playing with poop his whole life. When he stopped working, he lived in a sterile environment. And I really think it's the loss of being exposed to these microbes that probably did him in. And in fact, he did die of Alzheimer's. And I probably would say it's probably the microbe he was culturing the whole time. So I think and then you look at Béchamp versus Pasteur. Pasteur lived till 71, Béchamp lived till 92. I don't know. I tend to what are they doing? What are those people? We should be looking at what are people that are living to 100, not say it's just their genetics, but say maybe there's a microbe in them. And by the way, we're looking at that. We've looked at centenaries. So that's why I'm confident to say here, it's not just genetics.
I was going to ask that. Yeah.
I'm fascinated by the concept of, which has just been recently sort of resuscitated out at Stanford, heterochronic parabiosis, where you take an old animal and a young animal and you sort of mate their blood supply so that they intermingle their blood. And what they find is that the older animal experiences substantial rejuvenation. The younger animal kind of gets the short end of the stick and ends up having all sorts of issues and accelerated aging. Question is, well, okay, what's in that's what's in the blood supply of the young or in the supply of the old that's causing these things in the partner? But what about doing this with younger and older animals and doing, instead of parabiosis, you're doing or equivalent parabiosis with the microbiome? You take the microbiome out of a young, healthy 6-month-old mouse and put it into a 24-month-old mouse or 23-month-old mouse. Do you see some level of rejuvenation in the older animal? Because that's something that you wouldn't necessarily have to do, damage to the younger animal. You could just culture it, proliferate it and put it into the older animal.
I mean, certainly the research needs to be there, right? But I can tell you that in the microbiome, there's the component of the colonic mucosa. Mm-hmm. That's also, so when you think about fecal transplant, right, and implantation, you have two components. One is the microbiome. The next one is the colonic mucosa, right? And we see that when we do fecal transplant, where you could put a younger person, you could put a good microbiome, but it's not implanting. The mucosa is just destroyed. There is a point, and I think that's going to be the future studies to look at, do we restore? But we're so far from that, because first we have to isolate the microbiome. We have to kind of look at, the colonic mucosa, biopsies. There's so much research needs to be done on that to get to that answer. But, there is a component of it's just not engrafting. And is it not engrafting because the colonic mucosa changed to adapt to accepting new microbes, right? So think of your newborn baby. He's exposed to breast milk from mom, no stress in life, no alcohol, nothing. He's happy, non-anxious. He's got a lot of bifidobacteria because he's in that perfect bubble. But then he needs to survive and he goes to kindergarten and now he's exposed to all these microbes. The mucosa, the microbes change to adapt to that environment, to that stress. Now he gets a job, he goes to college, he gets a job, he gets married, he has kids. That transformation, that change again continues. So unless you're kind of following that change, you don't know. I propose to say that these young microbes are not going to engraft in an old microbes. And I think it's partly because the mucosa changed. And we see that with autism, right? Where there is a point where it's too late to fix these kids. They come in, they're 35 years old, autistic, and you could do as much implant as you want. You don't necessarily restore them back to the way they could have been. And that's because they've already adapted to the environment. The mucosa has already become what it's supposed to become. And you need a lot more. Those are the studies that need to be done. But that's definitely, to me, that's where we should be looking at, instead of pushing products that are just giving us 20% success on a disease. We should also be looking at, forget the microbes, but also the sound frequency, the waves, the music, the light, the foods we put in, right? Because all of that plays a role. I cannot increase a person's bifidobacteria if I don't control what they're putting in their mouths, the environment that they're in, and what they're doing to their bodies. Okay. Mm-hmm. Because if I'm giving them an implant, right, and I'm changing the microbiome, but then they're traveling, 36 hours on a plane and they're drinking alcohol, and they're partying like crazy. Well, they've just destroyed what I just was trying to grow, and I always refer the microbiome to gardening. In order to garden, you have to remove the weeds, plant the seeds, and let the seeds grow. If you give a hurricane or a tornado, the seeds are going to fly. If animals come in and eat the seeds, nothing's going to grow. So I think, that's what it needs. And unfortunately, a lot of people will come and say, Dr. Hazan, I want a fecal transplant. And I'm going to look at their environment. I'm going to say, you need to change this, this, and that. If they're not going to change those things, there's no point in me giving them a new microbiome, because they're going to pour gasoline on the microbiome, and I'm trying to put on the fire of their microbiome, and I'm trying to put water in there to stop the fire from propagating. And there is a point where you have killed so much by doing so much that you cannot restore. Okay. And I'm going to give the analogy of, you just nuke the whole city. Don't expect to grow tomatoes and fruit trees when the whole soil is destroyed, right? You have to let time and the planet and the microbes from under the soil pick up. So I think, unfortunately, we do too much. We do too much as doctors.
Yeah, we hypothesize, and JP wrote the article on this, that the autonomic nervous system has this important correlative function with the microbiome, with so many other inputs on the psychological side, on the physical side, on the movement, blood sugar side of things. And all of that is going to dictate whether we go into a more sympathetic dominant state or not. And the more we go into the sympathetic dominant state, the faster we're going to age. And I think there's a microbial signature that shows more resilience, more health, more support, and that terrain is one of health, of growth, and it's this rich environment. The analogy that I've kind of used for my patients, and I have this visual, and I'm the only person ever to link the Lion King movies to the gut microbiome. But if you remember when Scar was the king of the jungle or the king of the savannah, he caused complete chaos. The hyenas were ravenous, the grass was all gone, and the savannah became brown and it was barren. But when Simba took back over, they were able to restore, rebuild, because it was through balance and through creation of not one species overtaking and all of the species having a little bit of support, that they could create this healthy environment again, right? When Mufasa and Simba were the king, it was all about balance. It was creating an environment that was helpful for everyone. And I think that's ultimately the goal on the microbiome side, is to create that healthy environment that remains healthy as long as physically possible. Would that make sense?
You have an unhealthy knowledge of the Lion King.
I have two very young daughters.
My take on this is that you were just talking about the hallmarks of aging. And the hallmarks of aging are these symptoms that seem to appear in all people who are aging, and frankly, in all species eukaryotes that are aging. Dysbiosis is one of those hallmarks, but it is the one hallmark that actually is a statement not about you, but about your environment, because our microbiome is, in fact, the environment in which we live. It's external to us. Even in our airways and in our gastrointestinal tracts, it's the non-sterile exterior of the sterile interior of our bodies. And so I actually view dysbiosis as an environmental hazard, the same way ionizing radiation is, or the same way toxins are. It is an external thing that we need to have some regulation over. And it's interesting to me that there's this interface between our own genes allowing certain profiles of microbes to exist in our microbiome. And I think we need to understand what our genes allow and then restore what it should be. And it may be different for different people. It will be different for different people. So going back to something that you said earlier on, which was, is a fecal matter transplant going to be a cure-all for everything under the sun, all these neurodegenerative conditions you were talking about? Well, no, it's not. But is it going to be something that we need to be even more specific about determining what is the genetic profile of the patient? Then that will lead to the right transplant, because a transplant from you to me may work beautifully. It may work terribly. And your microbiome works wonderfly in you. It may not work in me.
Oh, 100%. I think my microbiome would kill more people because, they can't handle it. It's like,
It's that
hurricane.
Moroccan.
Yeah, it's a hurricane in motion. I wouldn't recommend it, but you're absolutely right. I think we all have different temperaments and we all have different microbiomes. And I think it's important to match. And here's the thing, there's a compatibility issue, right? You know how and I'm going to correlate this to, how there's sometimes people, it's funny because I always say I have a polarizing personalities. People either love me or they hate me. There's like no in between, right? But most people are kind of curious about what I do. But I think that's a compatibility issue, right? Think about when you look for a spouse, right? It's someone that has the opposite of what you have to bring you back into balance, right? I have two girlfriends that are like, both have disease, okay? One is constipated, the other one has diarrhea. And they both have different patterns of what they eat. They both have different symptoms completely, but they join together, they're best friends. And it's so funny because I look at them and I'm like, I think one of you is the best donor for the other and the other one is the best donor for the other, because it's so opposite, right? And yet they're so similar in their personalities in a way and best friends. So I think we are attracted to people that have what we're lacking, okay? We are
That's
also
That's
probably
immunologically.
holding. I think I'm going to push it even further to say hate is lost microbes. A person that hates is probably missing microbes from the person they hate. Okay. And you see that with when you see a couple and one patient is bipolar and the other one isn't. And you could see, wait, the person with bipolar disorder is missing all these microbes and the other person is not. Are they attracted to each other because of that lost microbe that one has? And is the other one hating because they're missing microbes that they should have had, right? So I always say your enemy is just your perceived enemy is just missing microbes. Once you understand that, you can start like reasoning with people to say, look, there's a bigger picture here, okay? We're all different. We all have our different traits. You hate me because I do this, and I hate you because you do that. But guess what? Somewhere in the middle, we can bring those two, ideas together and create a solution or evolve, right? And that's the key right now. Humanity needs to evolve. Humanity needs to evolve beyond the gender, beyond the race, beyond the religion, beyond the fights, beyond the money. It needs to evolve to understand the microbiome, okay? Because at the end of the day, listen, somebody offered me a lot of money for Progenabiome to shut me down. At the end of the day, that money is not going to save me. Trying to understand the microbiome is going to save me. Or at least, and I say trying because I don't think in my lifetime I'm going to figure it out. But trying to advance it, at least I'll have no regret to say I coulda, shoulda, woulda. Because imagine if I'm that person with Parkinson's or Alzheimer's, and now at least I'll have tried to change the path, to see another direction. I think that's where we all need as humanity to come together. Listen, autism to me, is the miracles from God because autism affects every population. It affects the people from Saudi Arabia. It affects the people from Morocco. It affects the people from India. It affects Americans. It affects everybody. It affects every religion, every gender. So we need to come together because here's the biggest crisis right now. 1 in 12 boys in California have autism. In 100 years from now, don't even ask yourself, it's going to be 1 in 1.
It's going to be.
And then once it's 1-on-1, humanity will stop. And we're not talking about autism like, oh, I'm on the spectrum and I can do multitasking, okay? We're talking about autism that cannot verbalize, okay? Are humans going to stop speaking? Are we going to communicate by, typing? So imagine restoring the microbiome and achieving speech in a kid that's nonverbal. That tells you directly microbes are helping speech, which is like the next level. What happens when you don't speak? So you can fight all you want and you can preach about your God, your religion, your race, your gender. But guess what? At the end of your political affiliation, people send me all sorts of crap on politics. I'm like, I don't want to hear about it. I'm all about the microbiome. Don't send me stuff that's going to distract me from my mission. I don't care. We're being influenced to hate because hate allows for marketing 101. It creates controversy. It ignites the person, when they bring up religion. Oh my God, both sides are heated. When they bring up, races, when they bring up color of skin, both sides are heated, the whites and the blacks, When they bring up women, women that stay home versus women that work versus men, versus men that stay home versus identity crisis, all of it. All to distract us, to sell a product and to sell fear. And ultimately, the person that's creating all that is going to be the patient, is going to have a kid with autism. And they really have to wake up in the morning and say, did I do the right thing? Sure, it's great that you've got all this money and you're flying in private jets. But guess what? If you're sick and paralyzed or your kid is autistic, it doesn't matter. It doesn't matter.
Yeah, I think this is
I
really
get heated
important
about
ultimately.
this.
It's fair enough, right? Like you're passionate.
I get so mad. Like the whole pandemic, I wanted to be like Noah and build my ark with microbes 2x2. Because I'm like this tube.
It's a lot of microbes.
We're not focusing on the big picture here. The big picture is we're dying. We're going extinct.
Yeah, this is causing it. Totally.
We're devolving.
And the planet's going extinct. Now, maybe on the big universe, that's what's supposed to happen. Who knows? And then another planet's going to restart. Who knows? But we're at an interesting, fork in the road right now where we can continue doing what we're doing and we're going extinct. Or we can stop and say, let's explore the microbiome. Let's explore the universe. Let's explore other planets. Let's explore humanity as a whole, I think that's what we should be focused on. What is the microbiome of Mars looking like, right? Of the planet of Mars compared to the planet Earth, right?
Interesting.
So I think it's all there.
I love it.
All the clues.
That's how Mark Watley, to quote a different movie, The Martian, that's how Mark Watley was able to grow his potatoes on Mars with a microbiome.
Totally.
So
yeah, they planted poop in Mars.
Yep,
So
that's the way to do
hopefully
it.
they planted right poop.
It was full of bifido.
No, maybe not.
Or not.
Maybe not.
This has been an awesome conversation. I've loved learning from you. JP, any final thoughts on your side?
No, I think it's such a huge topic. It's such a huge area to learn from. We are just at the earliest stages of it and trying to understand the microbiome and how human genetics plays a role in what microbiome each person has. This is such a fertile area, and it's not to throw a pun in there again.
It was a great pun.
But
and by the way, call to action. Let's see what the vagal nerve stimulation does.
100%.
Microbiome. Let's do that research.
I love that.
That has been an idea that has been floating around for at least a decade. Paul Durham down in Missouri. Let's get on it. Let's do it. So
let's do it. Let's get some funding. Let's hope that somebody's listening on this show and wants to hear.
We'll send it to him directly.
I think there's a need for neurological to understand what it does,
totally,
especially in the neurological space.
Totally.
So
absolutely.
All right, guys.
Sabine, this was great. We'll put all your links in the bio, so anybody who wants to learn from Sabine, go ahead and click on those links in the bio. And thank you so much for joining us. For anybody who got to this point, share this episode with one person who you think could utilize this information to upgrade their health. And we'll catch you all on the next one.